Amaya is going to talk to us about Working Group 4, guidelines and SOPs. Amaya.
Thank you. So, well, I'm going to summarize the work performed in Working Group 4, which is the working group dealing with guidelines and SOPs. First, I'm going to give you some general information about this working group. Then I will talk about the different achievements and also about the projects that are still ongoing. The goal of Working Group 4 was the production of guidelines and SOPs for use of the hCOMET assay in biomonitoring, and this goal is related with the objective number 6 of hCOMET, which was to provide guidelines and SOPs for best practice in design of human population studies and in performance of the hCOMET assay, and to encourage the use of standard protocols to facilitate the comparison of results from different studies. But those are the people involved in Working Group 4, and as expected, we have collaborated very actively with the rest of the working groups. We had to achieve 2 milestones. The first one was draft guidelines for human studies sent to hCOMET partners for comments. And the second one, to collect data on individual protocols from database. We also had 2 deliverables, an optimized protocol for hCOMET assay, and a training course. Regarding the first milestone, we prepared and published in collaboration with people from other working groups a paper entitled Application of the hCOMET assay in Human Biomonitoring and hCOMET Perspective. This document is not a proper guideline, but it contains useful information to design and run a human biomonitoring study in which the hCOMET assay is going to be performed. It describes the effect of intrinsic and extrinsic factors on the level of DNA damage. It covers the inter-laboratory variation in levels of DNA damage and reproducibility. It includes the procedures to collect and storage human samples, showing the lack of standardization in these procedures. And it also deals with the statistical issues related to both sample size calculation and analysis of the results. And in this sense, the paper can be considered a guidance. Regarding the second milestone, the collection of data on individual protocols, we published another paper, also in collaboration with people from other working groups, entitled Technical Recommendations to Perform the Alkaline Standard and Enzyme Modified hCOMET assay in Human Biomonitoring Studies. In this paper, we include data from 55 protocols from the database prepared by Working Group 1. Well, to illustrate the differences in the methods in the protocols, here you can see the range of conditions or the range of parameters used for some of the steps of the hCOMET assay. The percentage of low melting point agarose goes from 0.5 to 1.5. The duration of lysis goes from 1 to 2 weeks. The duration of unwinding goes from 15 to 40 minutes. The voltage per centimeter of electrophoresis goes from 0.7 to 8. And the duration of electrophoresis goes from 20 to 30. And those are just few examples of the differences. In this regard, the paper also contains a review about the effect of different parameters, such as the concentration of agarose, the duration of the cell lysis, the alkaline treatment, the conditions of the electrophoresis, and some others, in the outcome of the assay, showing that some of them has a big effect. And here we have 3 examples: the effect of the concentration of agarose, and the effect of the voltage gradient and the duration of the electrophoresis. As you can see, in treated cells, the higher the concentration of agarose is, here, less percentage DNA intake is obtained. In the case of the voltage and the electrophoresis time, the higher the voltage or the time is, more percentage DNA intake is obtained. All this highlights the need of a standardized protocol to decrease the interlaboratory variation. But finally, this paper includes technical recommendations about how to perform the standard and enzyme-modified COMET assay. And here we have just few examples. To keep a constant time of lysis, to use a constant volume of electrophoresis solution, to use a voltmeter to measure the voltage gradient across the platform, when using the enzyme-modified COMET assay to perform titration experiments to determine the enzyme concentration and incubation time, to include reference and standards in all the experiments, and to report the results obtained in the papers, and finally to code samples so that the scoring can be done blind. These are just some of the examples that we include in that paper. Regarding the first deliverable about an optimized protocol, working group, as it was mentioned by Peter, Working Group 4 was involved in the preparation of the protocol for the ring study. And it has been also involved in the planning and in the filming of videos about the different steps of the COMET assay to measure both DNA damage and DNA repair. And some of these videos are already available in a YouTube channel dedicated to the COMET assay. Actually, it's a YouTube channel of the International COMET Assay Working Group. And moreover, some of these videos are linked to the protocols that we are going to publish in the journal Nature Protocols. So far, talking about the publications in Nature Protocols, so far a protocol and a consensus statement has been accepted for publication in Nature Protocols. The protocol is about the COMET assay based, the protocol is about the COMET-based in vitro DNA repair assay that I think will be briefly explained in the next presentation, in the Working Group 5 presentation. And the consensus agreement is about the minimum information for reporting COMET assay, the MIRCA recommendations. And yeah, actually in these days, Peter Muller, who was in charge of this paper, has received the proofs. So we hope that it will be out very soon. Moreover, currently we are working on a third paper for Nature Protocol, which is a protocol about the COMET assay to measure DNA lesions. The pre-submission has been accepted, and we are now preparing the full paper. This paper involves many co-authors from hCOMET, but also many co-authors from outside hCOMET. Actually, we are now about 70 co-authors. And the number of co-authors is due to the extension of the paper. As you can see in this table, we are going to cover the use of different types of samples from different models or different organisms. So we are going to cover in vitro models, plants, non-mammalian animals such as crustacean, insects, airworms, mollusks, amphibian fish, and also mammalian animals such as rodents and humans. And moreover, we are also going to cover different modifications of the assay to detect other lesions than strong breaks and alkali labricides. And also, we are going to describe different high throughput versions, including the COMET chip, for instance. And we hope to have a first draft by the end of this month. So the people involved in this paper will receive some task very soon. We are also working on another paper about the collection and storage of blood samples for performing the COMET assay in future retrospective studies. And this paper is going to be produced together with Working Group 6 and Working Group 7, and has been divided in two sections, a literature review led by Peter Muller and Christian Delboux, and a protocol led by Eylul Banglu and Lisa Giovanelli. Well, regarding the deliverable, Working Group has participated in the organization and or in the running of 3 courses, the one that you can see in the slide. And actually, Andrew has talked about them before, so I'm not going to explain what were about. And apart from the papers that directly related to the milestones and deliverables, Working Group 4 has been involved in the preparation of other publications, as you can see in these slides, and also in the presentation of results in some conferences. Well, here we just have 2 examples. And moreover, several short-term scientific missions has been carried out in the framework of Working Group 4, on all of them related to modifications or standardization of the hCOMET assay protocol. And some of the results obtained in these short scientific missions are already published in the publications that I have presented before. And I think that's all. Thank you very much for your attention.